Hi everyone, it's Mr. Sinti, and today I'd like to discuss with you some of the more important principles of DNA sequencing. That's right, when you are studying molecular genetics or molecular biology in general, one of the very first steps is to extract DNA. But one of the things that you'd want to do with the DNA, one of the things is that you'd want to analyze it. You'd want to see what is the nitrogenous base sequence. And so you're interested in sequencing for a variety of reasons. And so this particular video is a brief look and an introductory video at that, at basic Sanger sequencing, which has been the type of sequencing we've been using in biotechnology for the past 30 years. And then it's going to briefly look at the next generation of sequencing, what we're currently using, although there's a little bit of an overlap. But in the last 10 years, next-gen sequencing has really revolutionized how DNA sequencing occurs. And so this is a video. Hopefully you'll enjoy it and learn something about sequencing. And so first thing right out of the gate is that, when you're talking about the complete set of genes of an organism, you're talking about the genomic sequence. So the whole genome of an organism is what we're talking about. You could specifically sequence particular genes or the whole set, which is the genomic DNA. And so before we get into Sanger sequencing in general, I just wanted to provide a little bit of background for those of you who are unfamiliar with gel electrophoresis, because this is an important principle in sequencing, and it's just an important principle in general when you're analyzing DNA. So one of the things about gel electrophoresis, it means that you're creating a gel, usually a sugar-based gel like agarose or polyacrylamide, and you're using an electrical current to separate, or electrophoresis, samples based on size of the fragments and by the charge of the molecules. And so you can see here's a vertical electrophoresis chamber, and this particular student is loading by a pipette, micropipetter, samples into this gelatinous material, which is soaking in a buffer solution. When the top is placed upon this, an electrical current will result, and it'll draw the DNA toward the cathode. And so this is a picture, schematic of that using a horizontal gel. And so if I were to animate this, if this was a sample of DNA right here, and you were running a gel electrophoresis, basically by pipetting samples into this well and you turn on the power supply, DNA is negatively charged, so it'll be attracted to the cathode, so it'll be moving in this direction right here. Now if you were to just pipette genomic DNA, since it's very large, it will migrate very, very little. And there would be very difficult to separate the difference between one genome and another, because they're basically really, really large molecules. So when you're analyzing through electrophoresis, you want to do something with the DNA before you put it into the gel, which is you would want to apply a restriction enzyme, which is, if you're unfamiliar with that, an enzyme that can cut DNA into various smaller fragments. So you would digest the DNA, whether it be a linear piece of DNA or a circular piece of DNA. Like, for example, what do you mean by that? Like, for example, a plasmid. If you were going to apply a restriction enzyme to this plasmid and it makes a cut there and a cut there, you wouldn't know it. Do you know, for example, if you had the sample like over here and you digested this, you'd have a small piece and a large piece, right? But you wouldn't know that. It sort of looks like just a clear liquid. You'd add loading dye so that you would see this. But basically what would happen is when you would run this gel, say you're going to load it in this first lane right here, this plasmid one right here. What would happen is that small piece right there would migrate further in the gel, because think of the gel as sort of a gelatinous, porous, kind of like Swiss cheese, kind of like a colander that can separate based on size. And so the smaller you are, the more you can sort of move through the gel, and the larger you are, you wouldn't be able to migrate very, very far at all. And so you'd be able to see where those restriction site locations were, because it creates a small piece, which is right there, and a larger piece, which is right there. Okay, so basically what we're doing is applying an electrical current to the gel, and that's causing the rapid movement of the DNA through the gel based on the electrical charge of the molecule. Now I mentioned the DNA is negatively charged, and so it's often loaded in a gel on the anode side so that it migrates towards the cathode. But the truth is, you really could use electrophoresis for proteins as well. And depending, if you were unfamiliar with the charge, you could put the wells in the middle and you can look at whether or not a molecule is negatively charged or positively charged if it migrates one way or the other. Or there's ways of turning your proteins and making them negative, depending on the buffer that you're using. If you're using something like lamelli buffer, you could do that if you're familiar with protein separation. Nevertheless, after you treat your DNA with a restriction enzyme, as I was mentioning before, you create fragments, so smaller pieces of DNA, and those can be separated by size. So in other words, if you were to digest that molecule and separate it, and you get a bunch of pieces like this, what you could say about this is that this is the smallest piece and this is the largest piece right there, and you can literally separate them using a gel like this, and so that's pretty useful. So this particular diagram is useful, I find. So if you were to load this sample here and the sample here and the sample here, you'll notice that when you turn the power supply on and the DNA starts to migrate through the gel, it'll begin to separate, and the smaller the pieces are, the further they'll be able to migrate through the gel. And then using a variety of staining techniques, you can actually visualize this on a gel. You can use ultraviolet light, or you could use some fluorescent dyes that will show up nicely as well. Okay, so you can compare shorter molecules with larger molecules, and I think that's essential in terms of understanding Sanger sequencing in particular, and that's what the video is about. I just wanted to show, this is an old sort of a retro photograph. It's actually if you don't recognize this, since it is kind of an older picture, it's actually a Polaroid picture. I pulled this out. This is a picture that I took in my lab many years ago using literally a Polaroid camera on top of the gel, so you can literally see these banding fragments here, and you can make some comparisons. Like, for example, just to give you an example, if I were to separate it like this and make it a little bit easier, you can see that this particular DNA has been digested. This is one type of DNA that's been digested by two different restriction enzymes, so it's cut a little bit different from one another. This DNA is different because it was cut, let me just call this enzyme 1, enzyme 2. This was cut, enzyme 1 cuts it in the same spot, but enzyme 2 cuts it a little bit differently. So you can see that this is the shortest piece here. And then this particular DNA is very similar to the first. And so this is the kind of a basic principle, but you can use digestion of DNA at particular sites with restriction enzymes to identify differences between individuals. And so this is something, if you're familiar with this, known as DNA fingerprinting. So that's pretty cool. So that's a useful forensic type of situation. And again, this is what it looks like when you're actually looking at it on top of the ultraviolet light illuminator. The bands using a particular dyes, something like athenium bromide, for example, causes the DNA to fluoresce like this, so it's pretty cool. So what can be said is these are smaller pieces. The shorter molecules are slowed down less, so they migrate further, and these are the bigger ones over here. So now that you have a little bit of a background on electrophoresis, I want to talk in particular about DNA sequencing and how the importance of sequencing is so important, because knowing the actual nitrogen-based sequence of a gene leads us to understand a lot more about how genes are controlled and how the genes are turned on or turned off in terms of gene expression, which is really important in terms of answering really fundamental questions about how cells differentiate and how they specialize during development. We might be able to crack some of those questions that we've had in biology for many years. And then Also, what's really important is in the field of evolution, when you're looking at phylogenetic relationships between organisms, no longer do you need to rely on phenotype and classifying. I mean, systematics has been based on the physical appearance and how closely related organisms are by a somewhat subjective criteria, if you will. I hope I don't offend anyone by saying that. But when you look at the genomes side by side between organisms, and the more similar the sequences are, the more closely related the organisms are. And it's fundamental, incredible evidence suggesting that all life on the planet is fundamentally descended from a common ancestor simply because the sequences of DNA that we've looked at so far indicate that. That's what the factual evidence suggests. Another thing that's really cool about sequencing DNA, and I mean, this sounds kind of science fiction-y, but it's not. It's like scientists at Penn State have reported that they've sequenced the whole genome of an extinct animal, like the woolly mammoth. That's pretty cool. So there's enough DNA remaining in the fossils and the bones that can be pulled out and sequenced. It's pretty interesting. And we're even interested in organisms, like in addition to human sequencing. My lab in particular is interested in coastal redwood sequence. We're conducting a study where we're comparing sequence from a variety of trees in a particular grove local to the lab, and our question is how genetically similar they are to one another. So we're looking at sort of a genetic diversity issue in terms of conserving those particular redwood trees. And so we're very curious about the nucleotide sequence of a particular gene, and we're using that as a means of identification in terms of how diverse the whole population is. And so all of this is made possible by a Nobel Prize winning scientist by the name of Fred Sanger, and this is a picture of him in the lab. The Sanger sequencing, as it's sometimes referred to, is what we've been using in biology, and again, it's still being used. We use the Sanger method in my lab when we're sequencing the redwood DNA, as I mentioned previously. It's particularly good for smaller sequences, but when it comes to whole genomes, the whole human genome process project that literally took a couple of decades to complete was done with the Sanger method. But now I'm going to briefly discuss the next generation of sequencing, which is so fast it's almost hard for me to imagine. But this is, again, Professor Sanger, professor at Stanford University, standing next to a gel. This happens to be a polyacrylamide gel, so PAGE, polyacrylamide gel electrophoresis. And so got to give you a little background about this Sanger sequencing a little bit. It's based on chain termination. And what that means is if you're familiar with DNA synthesis or DNA replication, DNA is constructed from these four basic nucleotides. They're called DNTPs. So deoxyribonucleotide triphosphate, but it's like adenine, thymine, guanine, and cytosine. So the normal DNTP has a hydroxyl group, and this is important, sorry for the detail, but it has a hydroxyl group in the 3' of the sugar deoxyribose. Now, if you're familiar with replication, which I hope you are, you'll know that that's particularly important because you need that hydroxyl group to extend the very next nucleotide. So the polymerization wouldn't occur unless you had that hydroxyl group there. So the enzyme that catalyzes the elongation of DNA, in other words, the synthesis of DNA, is a very important enzyme called DNA polymerase. I hope you're familiar with that. The Sanger method is based on termination of synthesis, and how it's terminated is that you have a synthesized, like abnormal nucleotide. And what I mean by that is the 3' does not have the normal hydroxyl group. So if this was in a tube and DNA was elongating or growing inside the tube, very similar to polymerase chain reaction, if you're familiar with that. I'll go into it in a moment. But as it's extending, if this defective, this is you're not going to be able to add any more nucleotides to it. So once this base has been incorporated, termination ceases. And so it's sort of the big terminator. And what Sanger thought he might do is tag this D. It's sort of like this is the one that's going to stop the termination, or actually cause the termination to occur. He wanted to label that using radioactivity. And so that's important in terms of like noticing what's going on in the reaction. And so this is what the DNTPs come when you order these in the mail. They're kind of like a clear liquid. But this is your D TTP and your D CTP, and then here's GTP, and then D ATP, like this. Okay, so these are the normal ones. Okay, by normal, what do I mean by that? The normal, I mean this guy. So you need to have normal normal nucleotides in the sequencing. Okay, so let's talk a little bit about normal sequencing, because when I talk about the inclusion of these special di-deoxynucleotides, these are the defective ones, the di-dioxy ones, and the reaction will stop the elongation from occurring. So if you're familiar with your double-stranded polynucleotide DNA like this, if this is your template strand that you wanted to sequence, first thing you'd want to do is heat it so that you'd open it up so it'd be single-stranded. Okay, so then you'd open it up like this. And if you're familiar with replication, you know that DNA polymerase isn't going to be able to replicate unless a primer is already put down. Okay, and so there's your primer right there. And so this is going to be in the growing 3' direction. So there's a hydroxyl group right there. And so this is normal. So what's going to happen is, I'll try to emphasize this with another color. The normal DNTPs are going to come in, and there's going to be extension like this. But then when in the tube, when you get one of those defective ones, so the DD, say TTP, comes in, and when that one's put in, say that's a thymine right there, so this would be adenine. If you put this one in, that's it. That means things are going to, and I'll emphasize this in red, things are going to stop, because that defective nucleotide is not going to have the 3' hydroxyl group, and so it's going to terminate the elongation. Okay, so these di-deoxynucleotides can be marked either using radioactivity, which is what Sanger used initially in the 70s and even in the 80s, but more recently we've been using fluorescent tags, which are a lot safer and a little bit easier to see as well, and I'll explain. So these fluorescently tagged big di terminators are going to incorporate themselves in, and you're like, well, what's the point of all of this? Well, first off, you need to have your template. So this is your DNA that you want to sequence right here in blue. Okay, so what you have to do is you need a primer. So you need to know a little bit about the area that you're going to be sequencing upstream of the unknown area. So you create a primer that will anneal or attach to this particular area. So this is just sort of like normal PCR if you're interested in amplifying a piece of DNA. So a primer attaches, and then DNA polymerase needs to be in the tube. So this is all in a tube right here. This is your template DNA in there. Then you got to put your primer in there. Then you have to add your DNTPs. These are the normal ones. And then you want to add DNA polymerase, which is going to extend the molecule. You're going to need to apply heat to open the molecule. Then you're going to want to cool it to allow the primer to anneal. Then you're going to want to warm it for DNA polymerase to extend. Those are the 3 phases of PCR. But here's the difference. Like, once you do this, then what you're going to then add is, in one of the tubes, you're going to add one of the defective adenines. And then in another tube, you're going to add one of the defective cytosine and defective thymine and defective guanine. And remember, these are the ones that don't have the hydroxyl group. So DNA would not be able to extend at that point. And so let me take a break from the, let's see if I can, take a break from this and show you a little bit about some video that I found that is particularly useful on this. So you can see the animation of this.
genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands.
Okay, so you have to heat it.
The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain.
Okay, let me just pause it there. So, in the tube, when you've added your template DNA that you do not know, say you added in the tube the DDGTP. So anywhere on here where there was a cytosine, anywhere on the original template there's a cytosine, because remember we're looking at the complementary. So if there's a cytosine, a guanine is going to be added to that, but then no longer. So replication will take place, but anytime a DDGTP is added, that is when termination occurs. And so you're going to get fragments of different sizes. Okay, so we talked a little bit about gel electrophoresis, so if you can predict how we might use this. So when you put it back together like that, you can see where all the
This
guanines
process
occur.
is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
So I find that to be particularly cool, but again, just staying with this, let me sort of just go with this. So again, I was mentioning in my lab we do sequence of redwood genes, and we ligate them into bacterial plasmids, and then we clone them in bacteria. And so then we go about purifying the plasmid and sequencing the plasmid. So this is going to show you a little animation of how sequencing a plasmid takes place. So let me push this here. So here's the area of interest. Now, I mentioned that this is an area that we don't know, and this is an area we do know, because if we know the plasmid that we're ligating our unknown gene in, we're able to construct a primer over in this area. Okay, so a primer occurs. So then we need to, here's the plasmid DNA and the target. Then of course we need to, heat the DNA. So we take it up in a thermal cycler like this, and we can, again, look at one particular strand, and we heat it, and then we will allow the primer to anneal right here. And then we're able to add DNA polymerase and DNTPs, and so it's elongating. So that's good, it's extending. So it's sort of like polymerase chain reaction. Then, look at that. Now we added a DDTP. And so since that has no hydroxyl group right over here, DNA polymerase cannot extend. And so this fragment is now terminated at this point. So we know that the very end of this fragment is a thymine, especially if we've fluorescently tagged it. That's important. So we fluorescently tag that. And so check this out. And so then we heat it, and then that gets removed again, and then we repeat. Okay? And this keeps going over and over again because it's random. It's like maybe the next time we do this, that particular endpoint wasn't going to be thymine, it was going to be further along. And so we keep doing this over and over again, okay? So let me show you this. This is another animation I think is particularly useful.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form.
Okay, through heating.
A mixture containing the single-stranded DNA, DNA polymerase, the 4-deoxyribonucleotides A, T, G, and C, and a single short primer is prepared.
So this is all normal PCR.
The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube.
Okay, so those are the dideoxy ones, are the ones that are fluorescently tagged with a defective hydroxyl group.
When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type.
Okay, so that's important. So those oligonucleotides are like these small fragments that can be separated by the gel.
The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
And then this is particularly important. When you load this in the gel, check this out. The DNA will migrate out like this, okay? And so these are small pieces, right? And you can tell that the very smallest piece of all migrated the furthest, okay? So it migrated the furthest. Look at this, it's only 1 nucleotide. And that happens to be A. And then the next one up, the next smallest one is G. So this one is comprised of AG. And then this one is C. And so as you can see, these pieces, this one right here is this. This is the longest piece, biggest piece, and the shortest piece. And you can literally go right down this and go, it's C, C, T, A, G, C, C, G, A. And this is how Sanger sequenced it. And so using that, hopefully that was helpful. So when you get back to this, you can see that these oligonucleotide sequences, when you load them into the gel, and each lane representing a different NTP, you'll see that you can literally go up and read this. Like, you know, even though it says, like from up here, that this is the smallest piece down here is in lane C. That means that's what you're reading, but that means that the actual DNA is a G, because we're looking at complementary. This is what you need to be able to deduce. And then the next one's G, so it's C, and then that one's A, so it's T. Do you follow? And so you can determine the sequence this way. And so it's somewhat tedious, because do you notice here it says manually sequencing using radioactivity? And so when labs did this, they had to literally make a photocopy of this and literally go with their Sharpie marker and go, all right, here's a C, there's an A, there's a G, there's a C. And so it was very impressive that you could sequence DNA, but it was very slow. And so when you put it all together like this, you can see, again, it's just like PCR. You add your primer, then you add your normal sort of PCR ingredients, your magnesium chloride, I've not gone into that, buffer, DNTPs, but then you add these DD nucleotides, and those chain terminate the process. And so you can literally, again, this is using a polyacrylamide gel, electrophoresis, or PAGE. You can literally go, okay, there it is, C, G, and then there's an A, and then there's a T, and you can move it up. And so the order of the fragments of the gel are interpreted as the nucleotide sequence when you go from smallest to largest. Pretty cool. And again, here's a picture of the vertical polyacrylamide gels that were used, and here's the power supply. But lately I mentioned we're using reagents that are fluorescently tagged, like from Applied Biosystems. They have big dye terminators. And so these particular 4 nucleotides, these are the ones that we use in our lab, these particular nucleotides are fluorescently tagged. It's the same principle, but it's a lot safer. So you do the same extension, you put in your DNA that you're interested in sequencing, you put in your polymerase, your primer, your normal DNTPs, and then these specially fluorescently tagged ones that have an abnormal hydroxyl group over here. And so now, and it started off as 16 samples could be done at the same time, and then it was determined that instead of using these big gels like this, what you could do is you could make capillary tubes that are porous, and you can do like 16 of these, and you can eventually put 96 samples through this. It sort of looks like a big ship with these sails right here, just keeping the capillary tubes organized. But basically samples are drawn up from a well like this, are drawn up like this, and you like that, you put a plate on this, and this is done automatically, and here you have your 96 plate, and then all of your samples are in here. And what happens is the liquid or these DNA fragments are then drawn up through these capillary tubes, and it's basically electrophoresis. You're separating them based on size. But what's interesting is there's a laser over here that interprets the fluorescence. And so this is what it looks like when you put the capillary tubes into the whole machine. Here are the samples are drawn up, they go through the tubes, and then laser reads the fluorescence. And so this is a sort of a schematic of that. And you can see here, these are the shortest pieces and the longest pieces like this. And so when they go through, the laser detects the color. And so then it goes into the computer, and the computer will actually allow you to see this on the screen or print it out. And you can actually see in this this printout right here, the actual DNA sequence using the fluorescent tags. And again, this is separation by capillary electrophoresis, because these tubes are capillaries. They're very thin, and then a laser reads it. And again, the fragments are created because at each endpoint, the dye is added, and it arrests termination at that point. So whether you're using radioactivity or you're using fluorescent dyes, it's going to give you the same readout, but it's a lot safer with the fluorescence. And again, here's a couple of scientists working with polyacrylamide gel electrophoresis using the capillary method or Sanger sequencing. I thought I'd throw in this chromatogram. This is what the readout looks like. This is a small portion of the gene that we're studying in my particular lab. We were looking at glyceraldehyde phosphate dehydrogenase in my lab. This is a portion of that gene sequence in Sequoia. So it's pretty neat. So you can actually read the sequence out. But first you have to extract the DNA. Now let's talk briefly about this next generation sequencing. Sometimes it's referred to as next gen. It's kind of hip. There's a couple of different ways of doing this. I'm just going to briefly discuss one or two ways, just to give you just an idea of what we're talking about in terms of speed and power of this next generation sequencing. It staggers the mind. Like, yesteryear, doesn't seem that long ago for me, 1995, we're able to accomplish 200 base pairs in a day. Like, pretty impressive. And then in 2001, 3,600 base pairs in a day. Again, Sanger sequencing. We're now able to sequence 1 billion base pairs per day. So we're talking that the whole complete human genome is around 3,000 base pairs. So we're talking 3 machines can do this in a day, which took decades. The Human Genome Project to sequence the human DNA took decades. It can be now accomplished in 1 day or 3 days if you have one machine. So in the last 10 years, this is basically again, hyperbole alert, a revolution in biology. Revolution, next-gen sequencing. Most important. It doesn't rely on chain termination, though. It's not Sanger sequence in nature. And so I just want to throw out one of the ways in which this is like the future of biology, where we're basically just taking a whole complete DNA that's extracted and basically feeding it through a pore, like you'd found in a channel, a protein channel in a plasma membrane. You can create artificial ones. And basically, as the DNA migrates through that channel, you'll be able to detect small pH changes in the sample as each nucleotide travels through. There's a slight fluctuation, and so we'll be able to just simply send it through. It's pretty wild. But most next-gen sequencing works a little bit like this. We would take DNA and we'd extract it, and then we take the genomic DNA and we amplify it, and then we fracture it into smaller pieces, and then we try to take each of the fragments and attach it onto a bead and basically amplify it using techniques such as this normal PCR. And so you get all these pieces sticking out of a bead. I know that sounds a little bit weird, but these are millions and millions, again, of copies of DNA that you're trying to sequence. And then you put these beads with all this DNA sort of sticking out on it into basically a slide with these microscopic wells. Okay, so you put the DNA in there, and then what happens is robotics comes along and it adds some primer to this. Okay, so it sticks to this piece right here, the little primer, and then the solution with the 4 nucleotides is then added to the well and then washed, and then another one is added and then washed. And so basically you're sequencing by synthesis. In other words, as the new DNA gets added, let me show you what would be happening in one of the samples. So if here's your DNA that you're interested in sequencing right here. So here's the primer, it attaches. Here's DNA polymerase. And so, okay, what's the very first nucleotide that's going to attach to the T? It's going to be an A. So if you added this nucleotide to this and it attaches, the computer is so sensitive that, if you're familiar with sequencing, when you add a DNTP, a pyrophosphate is removed during the synthesis, because 2 of the phosphates need to be removed to provide the energy necessary to catalyze the phosphodiester bond of the new phosphate group to the sugar. And so therefore, just that slight detection of light flash is then recorded. So there's like an illumination if when you're adding adenine and it's the correct base, it'll slightly illuminate, and that'll be detected by the computer. Over here, if you add, for example, if the next base is, do you see how it's G? So if you added a C, it would illuminate, but you didn't add a C, you added a T, and so, ooh, it doesn't shine. Okay, and then the next one, it's like, and let me erase this to make sure. So, and then, okay, so the correct one would be a C, and you're like, oh, but I did not add a C, I added the G, so it doesn't shine. And then, hey, look, I added the C, and it does shine. And so the pyrophosphate causes on the screen little tiny lights to blink up, and the computers are very advanced, and they can simply read this, and we're talking about incredible speed. And so the future is now. I mean, I'm sure they were thinking this years ago, but I'm telling you, like, this is the era that we've been waiting for in molecular genetics, which is the era of personal genomics. In other words, we're not just sequencing a person's, any person's DNA. We're capable of sequencing individual people's whole genomes quickly, within a day or 2 or 3. Now, you're like, well, why would we be interested in this? Well, I mean, we're talking about customizing medicine. So we want to know whether or not this particular medicine is going to be useful for that particular person. Maybe there's a side effect if we use it, maybe there's not. And so, and also, more importantly, to identify specific disease genes in a person. And therefore, things could be perhaps someday fixed, or at least anticipated in terms of the person's outcome in the future. And there's companies that are making some profit on this. There's a company called 23andMe, where you can literally, they'll send you a box like this at home, and you can really take a cheek swab and send in your DNA as your template, and they'll be able to send back sequence, and they'll be able to tell you whether or not you're going to have a particular disorder or not, or if you're a carrier for it, or what are your chances. It's pretty remarkable. And so these are some wild times. It's pretty surprising and humbling all at the same time. Like the human genome, one of the things that we were like, yay, 2003, we have the complete human genome. But we were kind of like, chin in the chest a little bit. We're like, wow, we only have around 35,000 genes. That's far less than we maybe anticipated. And it's only just a few times more than other smaller things like fruit flies and nematodes. And it's like, well, gee, I thought you said that DNA for humans had, 3 billion base pairs. But as it turns out, we have a lot of non-coding DNA called introns. And a lot of repetitive sequences. So even our most powerful computers are having a difficult time completely organizing where those sequences, since they're repetitive, where they actually are located within the human genome. But these next-gen sequencing is going to change this, and we're going to be able to figure this stuff out pretty remarkably. And so I mentioned the woolly mammoth, and so there's There's thousands of different bacteria that have been whole genome sequenced. There's been hundreds of eukaryotic organisms that have been completely sequenced, and there's thousands slated to be sequenced. And so as fast as I'm being able to produce this video, I'm sure it's going to be outdated in just a few years. And so stay tuned for the next update on next-gen sequencing. I hope this was helpful, and I hope it stirred some curiosity about DNA sequencing. I hope you enjoyed it. Thanks for watching.